Immunochemical screening by using bacteriophage lambda GT11 vectors or even lambda ZAP vector, which is a type of phase-mid vector, they can also be used for immunochemical screening. The previous immunological method of Broom and Gilbert, it was based on the library construction of the plasmid. So the library was on plasmid recombinant molecules. But here the library construction can be accomplished by using bacteriophage lambda vectors like lambda GT11 or lambda ZAP vector. They may be a type of insertional vector. So one of the advantage is that here the larger DNA fragments can be incorporated, and the in vitro packaging, it is very efficient, so that as compared to plasmid libraries, we can get high number of recombinant phages. So initially this technique was used in late 1980s, and here the cDNA libraries were constructed, and the cDNA libraries, they produce fusion proteins along with beta galactosidase protein. So fusion proteins were produced and they were then detected. So this method has the advantage that as compared to plasmid vectors, so large number of recombinant phages are there. And initially this method was evaluated with lysogenized phage particles. So their induction was performed, and then plaque formation was there, and then it was detected by using specific antibodies. But later on, the direct phage method was used, where recombinant phages were used, and then the cell lysis is there by using appropriate host organism. तो बैक्टीरियोफेज लैम्डा GT11 या लैम्डा जैप में जब लाइब्रेरी कंस्ट्रक्ट की गई, तो इसमें जो होस्ट ऑर्गेनिज्म यूज़ किया गया वो था लैम्डा 1090 आई थिंक. तो ये इकोलाई की एक स्ट्रेन है जो कि होस्ट ऑर्गेनिज्म के तौर पर यूज़ की जा सकती है. तो इसका एक एडवांटेज ये है कि इसमें लैक रिप्रेसर होता है जो कि हाई एक्सप्रेशन होता है, जो टारगेट जीन का उसको प्रिवेंट करता है, क्योंकि हाई एक्सप्रेशन जो है sometime if it is untimely तो it may be hazardous. तो जो एक्सप्रेशन है जीन का ये उस वक्त इंड्यूस होता है when we add some sort of inducer like IPTG. It is a inducer that can be used to induce the expression of the particular gene. जब जीन एक्सप्रेस करता है तो होस्ट ऑर्गेनिज्म की लाइसिस करते हैं, तो जो लाइस प्रोडक्ट है या एक्सप्रेस प्रोडक्ट है इसको डिटेक्ट किया जाता है by using specific type of antibodies. So one of the advantage of this technique is that the replica plating it is not required as in case of the previous method. This is the basic outline of the immunochemical screening by using lambda GT11 and lambda ZAP recombinant plaques. As I told you that here we can use different type of lambda insertion vectors. Here first example is lambda GT11 and lambda ZAP vector can be used. Here the cloned gene, it can be expressed as a fusion protein because it can be inserted within lambda Z gene. So lambda Z gene along with target protein, it can be expressed. So in polylinked site in case of lambda Z, the target DNA it can be inserted, and it can be digested with EcoR1 in case of lambda GT11. So we have inserted cDNA here in case of lambda GT11 or lambda ZAP vector. And then this titer of bacteriophage lambda, then it can be plated on a host organism, and here host organism it is E. coli Y1090. So the phage particles, they are incubated here for 4 to 6 hours at 37 degrees Celsius, so that the plaque formation it is there, host cell lysis is there. After host cell lysis, the nitrocellulose membrane it is placed on the plaques. So here replica plating it is not required because the master plate it is retained after the plaque lift. So sheet, this nitrocellulose sheet it is soaked with IPTG inducer. So when it is placed on the lysed colonies, now the expression it is there of the target protein. Now after expression, the nitrocellulose sheet it is lifted, and then the expression product it is detected by using specific type of antibodies. And here the master plate it is retained here, and if we have the idea that these plaques they are recombinants, then we can process those plaques further. So this is the basic screening strategies where we can use lambda GT11 vector or lambda ZAP vector for a screening expression product. Now you have the idea that in original method, radiolabeled or iodinated antibodies have been used. Now in this method, this radiolabeled antibodies technique it can be superseded with other methods where we can use non-radiolabeled antibodies. For example, here the antibodies like the secondary antibodies, they can be coated with certain chemical agents that can be detected later on. And in some cases we can also use polyclonal antibodies for screening. Polyclonal antibodies are those antibodies that are produced by different B cells, and they can react with different determinants on the target protein. So the specificity of the polyclonal antibodies it is high as compared to monoclonal antibodies that can react with only single determinant.